Okay, in here, they skip something, crossover, genetic recombination, and then independent assortment. Yeah, they skip something here. They only talk about the prophase 1, and then they skip to meiosis 2, which is not quite make sense, because they skip another important event that occurred in metaphase 1 that also increased genetic diversity, but they will talk about this in the next few slides. So what happened in the meiosis 2? Meiosis 2 is basically the same as mitosis in that it is the separation of the sister chromatids. So there's not much to talk about if you understand mitosis. It is exactly the same as mitosis. But because in meiosis, at the end of meiosis 1, we have 2 daughter cells formed at the end of meiosis 1. Therefore, in prophase 2, you have 2 cells, you start with 2 cells, and then each cell go through meiosis 2, that means mitosis. Then at the end, you have 4 daughter cells. So that's the summary of the meiosis 2. So cell division, the whole purpose of it is to create genetic variation, genetic diversity. So it is, the reason is that when you have genetic diversity, genetic variation, then it is good for the offspring to have survival or for evolution. In other words, the best partner, the best mate you can get is not from the same origin. The best partner you can get should be someone that is totally different from your genetic makeup, meaning that interracial relationship is highly encouraged for the sake of genetic variation, to increase the genetic diversity of your offspring. Usually cross-racial relationship, the offspring, they are healthy, smarter, and so on. The reason is that the genetic makeup of one parent is totally different from the genetic makeup of another parent, so that the recessive gene or the disease-carrying gene can all be covered up. Mutation, of course, is another major source of genetic variation, but I can say that most of the mutation occur without having an obvious effect. Most of the mutation occur is a silent one. They make something that is not useful, not harmful, not beneficial. Only a few mutations that is actually harmful, and even a few mutations is actually beneficial. We'll talk a lot more about mutation when we talk about genetics later on. And we will talk more about alleles. Allele is basically a pair of genes. They can be homozygous dominant, heterozygous dominant, or homozygous recessive. Again, we will talk more about alleles in genetics later on. And through meiosis and fertilization, sexual reproduction can combine a new allele to increase diversity. What does it mean? Remember, fertilization is basically you have millions of sperm competing with one egg. But the million of sperm, each sperm in the millions, they have different genetic makeup because of meiosis. We'll talk more about what causes the genetic variation in the sperm. Even in the million, you could not find a single identical pair of the sperm. So million of different sperm, they compete with one egg. This is what we call random fertilization. Random fertilization will also increase the genetic variation in the offspring. So we briefly talked about this. In the prophase 1, your paternal chromosome and maternal chromosome, they are touching and grinding each other. We call it crossover. The transferring of the genetic material between your homologous pair increases the genetic diversity. We call this crossover, aka recombination. The reason why it is increasing the diversity, as you can see this picture, after the crossover, you can have 1, 2, 3, 4 types of cell. But try to imagine without the crossing over to occur, then you only have one maternal chromosome and then another identical maternal chromosome. And then you have one paternal chromosome and then another identical paternal chromosome. So you only have 2 types of cell. But with crossing over, you can have 4 different types of cell. And I can tell you that crossing over is not just exchanging one gene. When crossing over occurs, multiple genes can jump around. So it's not like just one here. You may have another blue paternal gene in this part. You may have another red paternal gene in this part, or you may have another red maternal gene in this part. So it can be multiple gene exchange during the crossover. That will further increase the diversity. Then the second method to increase diversity is occur in the metaphase 1. Occur in the metaphase 1. So I want to give you another analogy. So remember the first analogy. Okay, your parents, they are touching, grinding each other, and then they exchange body fluid, and then what happened next? When I asked this question to my other community college student, one of the students is very naive. She said that, oh, after that they hug. I said no. They kiss each other. I said no. And they said, well, you're too nice. Okay, this is not American. They are not like this. After they have sex, what will happen? I told them that, okay, after they have sex, they have something called post-orgasmic disgust, and then after that they will get divorced. Yeah, because they hate each other after sex. So this is what happened. After they touching and grinding each other in prophase 1, exchanging body fluid, they get sick of each other and they get a divorce. So what do they do when they get divorced? They go to a divorce lawyer. Usually, divorce lawyer, they will sit, it's a rectangular table, and then your mother sit on one side, your father sit on another side. But divorce lawyer, they are very crafty. In that they don't usually do one couple at a time. It's a waste of time. They usually ask a multiple couple. They who also want to get divorced sit together, so that there are different ways of sitting on the table. On the rectangular table, you can have all the wife sitting on one side, or all the husband sitting on one side, or you can randomly arrange, one husband sitting on this side, one wife sitting on this side, and then another husband sitting on the opposite side, another wife sitting on the other side, and so on and so forth. You can arrange it in different ways. The different arrangement of the husband and the wife will give different possible outcome. So similarly, in here, during metaphase 1, you can arrange all the maternal or mother chromosome on one side, all the father chromosome on one side. In this case, during the this is the meiosis one. At the end of meiosis one, you have one cell that are all maternal, one cell that are all paternal. And then at the end of the meiosis 2, you have 4 cells. 2 cells they are all paternal, maternal, and then 2 cells they are all paternal from father. Or you can arrange the homologous pair like this, so that after the meiosis 1, this cell you have one maternal chromosome, one paternal chromosome. This one you have one paternal, one maternal. And then after the end of meiosis 2, you have 4 different cells actually. So you can see that by arranging the chromosome position during metaphase 1, then you can have increase in the genetic diversity of each of the daughter cell. So, in summary, let me sum up with this one. There are 3 processes that will increase genetic diversity. One is that crossing over or recombination occur in prophase 1. The second one is this. We call it independent assortment. I forgot to mention the name. This is called independent assortment, the divorce. The divorcing of your parent is the independent assortment. Independent assortment is the second process occur in the metaphase 1 to increase genetic variation. The third process that increases genetic variation is we call it random fertilization. It's something that I told you before. Millions of sperm compete with one egg, but the million of sperm each one has a different genetic makeup. So that are the three processes to increase genetic variation.